previous lecture we have gone through the basics in chromatography. Also we have discussed about the different aspects of low pressure column chromatography and high performance liquid chromatography. Now, in this lecture we are going to discuss one of the techniques which is widely used, it is ion exchange chromatography. Now, as you know, many of the biological molecules are charged molecules at a particular pH. So, they carry certain charge either positive or negative at a particular pH. Like for example, proteins will carry a positive charge below their pi value and will carry negative charge above their pi values, and this particular property of charge could be utilized for their separation. So, ion exchange chromatography involves separation of macromolecules and other molecules based on interaction between charged solute molecules and oppositely charged molecules covalently linked to a chromatographic matrix. Now, ion exchange is the most frequently used chromatographic technique for the separation and purification of proteins, polypeptides, nucleic acids and polynucleotides and other molecules which are charged. Now, the reason for the success of ion exchange chromatography is its widespread applicability, its high resolving power, its high capacity and the simplicity and controllability of the method. Before we go into details, I would like to show you on your screen in very simple way the mechanism and working of the ion exchange chromatography. So, please focus on your screen and let us understand this whole chromatography, ion exchange chromatography, and then we will go into the details. So, what happens is that first thing is you have a stationary phase. Now, this stationary phase which is particularly charged molecule or a charge, ionic charge which is immobilized onto a support matrix, say bead. Now, when you prepare your resin, then what happens is that your resin will carry a particular charge. It could be a negative charge or it could be a positive charge, and it is equilibrated with a particular buffer. So, what is your starting condition? The starting condition is, if I say this is your bead and this carries say particular charge, say positive charge, then at the starting point where you have equilibrated your resin, the positive charge will be interacting with the counter ions. So, if I say these are the counter ions which are surrounding the resin, the charge on the resin. So, that is the starting position. Now, second thing will be, so what will happen, how this whole separation takes place, that we will add the protein or a biomolecule. Now, when you add a biomolecule which are oppositely charged, that is here resin is positively charged. So, biomolecule which are negatively charged will bind to this. Now, they will replace the counter ions, and they can replace the counter ions because they will have high affinity for the so biomolecules, say protein. So, protein is bound here, and protein is those protein molecule which are carrying negative charge will bind in here. So, what you have is, if I show it here, it is something like this. If it is surface, if this is positive charge on resin, then negative charge will be binding, and this might be of any molecule which is present in here. All right. So, once protein is bound, then you have these protein molecules. If there is a mixture of protein molecules, then they will bind with different affinities depending on the charge. If they have more negative charge, they will bind with more affinity, if less, then they will bind with less affinity. Now, what you have to do? You have to elute your protein or take out your protein from the matrix, and that is to separate different analytes in a mixture, and this will be done based on the affinities actually. And here you will put an eluent high ionic strength eluent, which will replace the protein with those ionic species in the mobile phase. Now, what will happen? Depending on, as you will increase the concentration of eluent or ionic strength, like say you can add NaCl, and as you increase the concentration of NaCl, depending on their affinities of proteins, they will replace it. So, if protein has higher affinity, then you need higher concentration of NaCl. So, next step would be, that is elution. This was binding, and next step would be elution, where what you have done? You have eluted it to say NaCl or some other eluent. So, what will happen? This positive charge will now, it will be replaced by say chloride ion, and your protein is, you will get protein which is eluted from. Now, remember this protein, which will be like, it is a mixture, it will come out at different strengths of the salt. All right. Once this is done, so you might get different peaks in here which are there. Once it is done, then simply what you have to do, regenerate. So, again the simple buffer will be put in, and extra Cl or extra eluent will be washed off, and you will regenerate the column, which will be as the starting position, and so this resin could then be reused for another purification. So, this was in very simple way I tried to explain the mechanism of the ion exchange chromatography. Now, let us little bit see how the working of the chromatography is done. So, what is done here is that you have a column, and there is a stationary phase in here, filled in here, and which is say up to here there is a stationary phase. Now, in ion exchange column, and this contains resin, which we can say ion exchange resin here, and it carries a particular charge. All right. And like I said, it is in the form of beads actually. It is like these are beads which contain the stationary phase. This is a support matrix. All right. Now, what you will do is, there are two ways, like through peristaltic pump, as we were discussing in earlier lectures. You can do it by peristaltic pump, or you can manually load the sample. So, if I have to manually load the sample, then I can load manually the sample with a micro pipette. So, this micro pipette, I can load the sample, and this sample could be, in ion exchange you can have any volume of sample, as this is a displacement kind of chromatography, where the ions or ionic species will bind to the oppositely charged ion on the resin. Now, even what you can do is that you can put, say in the sample you can put the resin itself and let it bind in a beaker. So, that is also possible, that in a beaker you can bind it, and then you can load it onto the column, that can also be done. So, what happens is, once you have prepared your resin and you have loaded your sample will get in, and as sample gets in, what will happen? It will, as it gets in and gets in contact with the resin, the charges will be saturated. So, if resin is positively charged, negative charge biomolecule will bind to it and slowly saturate it, and as these beads which are in starting get saturated, the biomolecule will move in and it will bind to others. So, it depends on the capacity of this stationary phase, as how many ionic stationary phase it has. Once it is bound, then what is done, that you have to wash it, so that unbound very weakly binding things could be. So, you have a knob here which could be opened, and once it is and this could be collected, flow through could be collected. Flow through will contain biomolecules which are not bound to the resin, because they are of the same charge, and this could be then separated by another chromatography, ion exchange only, where it has oppositely charged stationary phase. Now, once you have done, then you will do washing. So, first is you have prepared the column, by equilibrating it with right buffer. Then second thing is you are binding your analyte, that is oppositely charged, and then you will elute. So, after binding, then elution has to be done, that will be done by certain ionic species which can replace your analyte, and as you increase the concentration depending on the affinities. So, what will happen if you have different affinities, as you elute, like say I can use 50 millimolar NaCl, I can use then 100 millimolar, 200 millimolar, and I can go up to 0.5 molar or 1 molar NaCl, and depending on the affinities you will get the peaks here which will be separated according to the affinities. So, less affinity will come first and higher affinity peak will come later on. Now, once you are done with this, you can regenerate the column as I have explained and again reuse for another purification step. So, this is how the whole working of the column is done. So, let us get into details. Actually, this was just to make you understand how this whole thing works. Now, let us get in the details of our discussion. Now, theory of ion exchange says that separation in ion exchange chromatography depends upon the reversible adsorption of charged solute molecules to immobilized ion exchange groups of opposite charge. Now, most ion exchange experiments are performed in 5 main stages, and these steps are, as I have explained also. First thing is, the first stage is equilibration, in which the ion exchanger is brought to a starting state in terms of pH and ionic strength. So, which allows the binding of the desired solute molecule. So, the exchanger groups are associated at this time with exchangeable counter ions. Now, these are usually simple anions or cations, it could be like H, OH minus or Cl minus or Na plus. So, sodium chloride or hydroxyl or H plus ions could be the exchanger ions. Now, second stage is sample application and adsorption, in which solute molecules carrying the appropriate charge will displace the counter ions and bind reversibly to the gel. So, unbound substances can be washed out from the exchanger bed using starting buffer, that is called extensive washing. Now, once that is done, in the third stage elution will take place. So, what is done? The substances are removed from the column by changing to elution conditions which are unfavorable for ionic bonding of the solute molecules or biomolecules. Now, this normally involves increasing the ionic strength of the eluting buffer or changing its pH. The fourth and fifth stages are the removal from the column of substances not eluted under the previous experimental conditions, that is you can use higher concentration of eluent, and then re-equilibration with the starting buffer for the next purification. So, these are the stages which I also explained in a very simple way that you have to go through when you are performing column chromatography, and this also shown in figure here, which is like first stage where counter ions are bound, that is starting, then your these molecules which needs to be separated are bound to the column, and one of those will bind as per the charge. Then your gradient ions will be utilized like NaCl or some other, which will elute as per the affinities, and you have to, maybe it could be increasing salt concentration is used, and then once this is done it will be regenerated. So, that is how it is a very simple mechanism of ion exchange chromatography. It is a very simple, very versatile technique to be used. Now, an ion exchanger consists of an insoluble matrix to which charge groups have been covalently bound, and these charge groups are associated with mobile counter. The charge groups are associated with mobile counter ions. Now, these counter ions can be reversibly exchanged with other ions of the same charge without altering the matrix. The matrix may be based on maybe inorganic compounds, synthetic resins, polysaccharides, etc., and the commonly used matrices could be like polystyrene, cellulose, agarose, dextran, and so on. Now, in ion exchange chromatography, there are two kinds of ion exchangers. One is called cation exchanger, and this depends on the charge they carry. So, the cation exchanger and anion exchanger. Now, cation exchanger are the ones which possess negative charge as they are exchanging the cations. So, they by themselves possess negative charge groups and will attract positively charged cations. These are also called acidic ion exchangers because their negative charge result from ionization of acidic groups. Then there are anion exchanger and they possess positively charged groups and will attract negative charged anions. These are also called basic ion exchangers as their positive charges result from the association of protons with basic groups. So, if you see these looks something like that, if anion exchanger and there is a cation exchanger. So, anion exchanger will exchange anions and cation exchanger will exchange cations, itself they are charged accordingly. This is positively charged and cation exchanger is negatively charged. Now, there are a lot of examples of commonly used ion exchangers, as you can see in this table. There are strong anion exchanger, weak anion exchanger, strong cation exchanger and weak cation exchanger. Some examples are of strong anion exchangers are trimethyl aminoethyl, which is also called TAM. There is diethyl 2-hydroxypropyl. These are strong anion exchanger. If you say weak anion exchanger, then aminoethyl and the diethyl aminoethyl DEAE, these are weak anion exchangers. Then there are strong cation exchangers like sulfo groups are strong cation exchangers, which is sulfomethyl or sulfopropyl. Likewise, carboxy weak cation exchangers include carboxy group exchangers like which contains carboxy group or carboxy methyl groups. So, these are some examples, there could be many more of weak and strong anion and cation exchangers. Now, when we say strong or weak, it refers to the extent of variation of ionization with pH and not really the strength of the binding. Strong ion exchangers are completely ionized over a wide pH, whereas with weak ion exchanger the degree of dissociation and thus exchange capacity varies much more markedly with pH. If you say some properties of what are the properties of strong or weak ion exchangers, strong ion exchangers, their sample loading capacity does not decrease at high or low pH values. They are due to loss of charge from the ion exchanger. So, they remain ionized at a wide range of pH. A very simple mechanism of interaction exists between the ion exchanger and the solute. Ion exchange experiments are more controllable since the charge characteristics of the media do not change with change in pH, and this makes strong exchanger ideal for working with data derived from electrophoresis titration curves, etc. Another important characteristic is capacity. Now, the capacity of an ion exchanger is a quantitative measure of its ability to take up exchangeable counterions and is therefore of major importance. Now, capacity may be expressed as total ionic capacity or available capacity or dynamic capacity. Now, total ionic capacity is the number of charged substituents groups per gram dry ion exchanger or per ml swollen gel. Total capacity can be measured by titration with a strong acid or base. Then there is actual amount of protein which can be bound to an ion exchanger under defined experimental condition, and that is referred to as available capacity for the gel. Now, if the defined capacity or defined conditions include the flow rate at which the gel was operated, then the amount bound is referred to as dynamic capacity for the ion exchanger. So, when flow rate is included, it becomes dynamic capacity for the ion exchanger. Available and dynamic capacities will depend on many factors and which includes say properties of the protein. Now, properties of the protein will include say molecular size and charge or pH relationships. Then properties of the ion exchanger will also affect this capacity like exclusion limit of the matrix, type and number of the charged substituents, etc. Then chosen experimental conditions like pH, ionic strength of the buffer, nature of the counter ion, flow rate, temperature, all these things will certainly all these factors will certainly affect the available and dynamic capacities of a particular experiment, a particular resin. Another important factor is porosity of matrix. Now, the porosity of the matrix is an important feature, because the charge groups are both inside and outside the matrix, and matrix is support material. So, because of this, the matrix also acts as a molecular sieve, because matrix a flow has to be maintained through the matrix. So, matrix is not in itself it is a mesh like structure, and there is a porosity here, and when you immobilize these ions, they will be all over the matrix. They could be totally exposed or they could be somewhat inside. Now, large molecules may be unable to penetrate the pores. It might happen that certain ions or certain charge groups may not be approachable by the large molecules, because they will not be able to reach, while the small molecules will be able to reach. So, capacity will be affected by the porosity also. So, many times when you are selecting the ion exchange matrix, you will also look for the porosity, and it is like when we say CMC 25 or some C25 50, it refers to the porosity, and accordingly you can choose the matrix for a particular size of the sample to be purified. Mesh size is also important, which determines porosity. Like ion exchangers will come in a variety of mesh size called variety of size called mesh size. Now, finer mesh size means an increased surface to volume ratio, and therefore, increased capacity and decreased time for exchange to occur for a given volume of the exchanger. On the other hand, fine mesh means slow flow rate, which can increase diffusional spreading. So, there are a whole lot of different mesh size are available, and accordingly one has to choose that mesh size. As we have explained in before, mechanism of ion exchange is thought to be composed of 5 distinct steps, and this is like a little different from the procedure we have discussed earlier. One is, the first step is diffusion of the ion to the exchanger surface. Now, this occurs very quickly in homogeneous solution. As you put your sample, the diffusion occurs. Then diffusion of the ion through the matrix structure of the exchanger to the exchange site actually, where it has to bind, and this is dependent upon the degree of cross linking of the exchanger and the concentration of the solution. This process controls the rate of whole ion exchange process. Then exchange of ions at the exchanger site. This is thought to occur instantaneously and to be an equilibrium process. So, what happens? This happens like when you have a particular ion resin, which is kind of interacting with a counter ion, and there analyte comes or a protein comes, then it is replaced, this will replace the counter ion as in here, and like in anion exchanger, similarly the analyte will replace the counter ion as it has higher affinity. The more highly charged the ionized molecules to be exchanged, the tighter it binds to the exchanger, and the less readily it is displaced by other ions. So, this depicts the affinity of a particular analyte for the exchanger. Then diffusion of the exchange ion through the exchanger to the surface, and the selective desorption by the eluent and diffusion of the molecules into the external eluent. So, all these steps happen one by one, and this is how the mechanism is unfolds of the ion exchange chromatography. Now, the selective desorption of the bound ion is achieved by, it could be simply pH or the concentration of a particular ion, that is ionic concentration could be used. So, this was mechanism of ion exchange chromatography. Now, selection of conditions for ion exchange purification depends on many factors. A wide variety of ion exchangers and adsorption desorption conditions are available, and one has to choose according to what kind of experiment they are doing. what is their analyte, like say pH stability, what is the charges at particular pH, all these things has to be taken care, and likewise buffers and eluent has to be or has to be selected. Now, the factors involved in making each choice are described here. So, if you say choice of matrix, so we are discussing those factors. For choice of matrix, for low and medium pressure ion exchange, you can use say cellulose, which could be fibrous, microgranular or beaded. It could, you can use agarose, dextran, polyvinyl, lot of other matrices are available. For high pressure ion exchange, you need strong ion exchanger matrices like silica based or other resins which have been made for high pressure chromatography. These matrices mainly differ in their particle size and pH range, and those could be selected accordingly. A lot of commercial resins are available for high performance liquid chromatography. Selection of functional group is another important factor. For example, anionic or cationic groups has to be selected. Now, if the material to be bound to the column have a single charge, then either positive or negative, so then choice is very clear. Now, problem comes in amphoteric materials like proteins which might have both positive and negative charge. So, pH stability has to be considered here. If a protein is more stable above its isoelectric point, then an anion exchanger should be chosen, and if it is stable below its isoelectric point, then a cation exchanger should be chosen, as the charge of the protein will be accordingly be at its above or below its pi value. For a weaker ionized substances which requires very low or high pH for ionization, a strong ion exchanger is used. However, for certain other substances, weak ion exchanger are preferred, because strong exchangers are often capable of distorting a molecule so much that the molecule might denature. So, accordingly one has to choose and see the conditions. If it is a very weakly ionized substance, certainly strong ion exchanger needs to be taken. Now, the most commonly used functionalities are weak ionogenic groups, and these are in common use, these are weak exchangers like diethyl aminoethyl, DEAE commonly known as. This group is usually used in anion exchanger to purify negatively charged proteins, while the carboxymethyl is another commonly used or frequently used cation exchanger for recovery of positively charged groups. So, depending on what kind of analyte you have, you can choose one of the exchanger, either cation or anion exchanger, and most of the time weak exchangers are utilized. Now, buffers used in ion exchange chromatography, again you have to be careful in here for successful ion exchange chromatography. Now, in ion exchange chromatography, buffers are chosen in order to minimize pH fluctuations and maximize the adsorbent capacity. Now, two important factors or rules has to be taken care of. One is, that is, charged form of buffer should not interfere with the ion exchange process, and we will discuss that. And then again another factor is temperature, which will affect the pK value of the buffer, and such that its buffering capacity will be different in purification carried out in the cold conditions. For example, tris buffer will have, if you set the pH in say room temperature and you carry it to the cold room conditions, then certainly change in buffer pH will take place, and that might hamper your protein purification. The commonly used buffers, this is a table gives you some commonly used buffer with cation and anion exchange chromatographies. For example, with cation exchange you can see acetic, citric, mass, phosphate, HAPIS buffers are used with cation exchangers, while with anion exchanger you can say alhistidine, imidazole, triethanol, amine, tris, and all these buffers are used with anion exchanger. So, you have to worry about that which kind of buffer which will not interfere with the particular kind of type of exchanger needs to be used. Now, selection of adsorption and elution pH is an important factor, and for protein adsorption on the matrix, a pH about 1 pH unit above or below of the pi of the target protein is chosen, while for protein elution 0.5 of a pH unit from its pi at an ionic strength of 0.5 molar NaCl conditions are chosen, and these things needs to be standardized. These are not hard and fast rule here. When you do your experiment, one has to standardize that how much salt concentration needs to be taken for elution, or if it is done through pH conditions, what changes in pH has to be done. Now, selection of adsorption and elution ionic strengths like I said needs to be standardized, needs to be taken care of, and one has to go through the experiment to do that. Some guidelines on the adsorption step, the highest ionic strength which will allow protein binding is used, while on elution step the lowest ionic strength is recommended. So, reason for this are as follows. So, if you have a buffer, then you should use a buffer of highest ionic strength when you are binding the protein, and when you are eluting, then you have to use the low ionic strength buffer. Only thing you have to take care that your protein remains stable in both conditions. If say at the low ionic strength, if it is not stable, then you have to avoid that. Now, if the ionic strength is too low on absorption, then the protein will bind too tightly and effective elution will be made difficult. So, that is why one has to use high ionic strength. Then keeping the ionic strength as high as possible on adsorption minimize the binding of unwanted contaminants. That is very important, that non-specific or unwanted contaminants will be avoided if high ionic strength is utilized. Conversely, keeping the ionic strength as low as possible on elution minimizes the elution of bound contaminants. So, you can get a better purification. Now, the strategy outlined above simplifies the elution step, and a salt concentration say of 20 to 50 millimolar is usually used during the absorption, and say up to 0.5 molar NaCl is used during elution process. This graph here figure shows that you can use something like, say if this is your pi value and your pH or range of stability for a protein is certain pH range, then that could be utilized for and this could be seen, and particular pH of stability could be used for purification. So, the net charge of a protein as a function of pH and showing the pH range for stability and for binding to cation and to anion exchanger. You can say it will bind accordingly as per its the pH, either it is pH is above or below. So, this protein would be coronated graft on an ion exchange chromatography using a buffer adjustable to pH 6.3 to 8.4. So, accordingly it has to be like if it is say if its anionic protein is anionic at particular pH where it is stable, then it has to be purified on anion exchanger, and like here 6.3 to 8.4 is the right pH that needs to be utilized. Now, choice of porosity and mesh size where we were talking about. Now, small molecules are best separated on matrices with a small pore size, whereas macromolecule will need large pore size. As I was discussing, many commercial resins they tell that by certain specifications that what could be the mesh size and porosity, and accordingly if it is a macromolecule you require to use a larger pore size, if it is a smaller molecule then a smaller pore size could be utilized. Selecting a mesh size is difficult and determined empirically, as a small mesh size improves resolution, but decreases flow rate. So, accordingly one has to choose the conditions. All right. So, this was like all different factors we have discussed regarding when you are attempting to do ion exchange chromatography, and what are the factors you have to take care, you have to accordingly design your ion exchange chromatography experiment. Now, if we see the simple procedure as I was explaining earlier in a very simple way, the procedure in ion exchange separation is quite straightforward. The first step to be done is you have to select the matrix. Once you have selected the matrix, the pre-treatment of matrix has to be done. Now, remember matrix will be available either in say powder form or in some solvent, say 30% ethanol. So, the matrix has to be in both ways, matrix has to be swollen, it has to be put in proper buffer which needs to be equilibrated, or you can start with water and then you can put it in buffer. So, what will be done? Like as you put the matrix in the buffer or suitable buffer or water, mostly you can start with water, but then you have to exchange it to the suitable buffer in a beaker. So, once you do that, then certain fines which are really not cannot be used, and they will be unnecessarily increasing the volume of the matrix, can be removed by decanting. Then you will allow, though in ion exchange swelling is not so important, but you will allow it to exchange with the buffer, and then it will wash it and equilibrate it to the proper buffer, and then if say it has to be converted to a correct counter ion, that will be done. So, matrix pre-treatment is the first stage of ion exchange separation. Once you have done that, then matrix has to be loaded onto the column. You can also load your sample before loading it onto the column, and there are various methods of adsorption. So, different adsorption methods could be, one could be batch adsorption. Now, batch adsorption is carried out in free solutions, but it has a lower efficiency. Like I said, you can do it in a beaker. It is ideally suited for initial treatment of large volumes of sample. Say if you have a liters of sample, and what you can do is a suitable amount of matrix could be directly put into the, or pre-treated matrix could be directly put into the this large volume of the sample, and you can gently stir the sample, and the protein or the analyte will bind to the stationary phase. Now, this does not suffer from problems of bad swelling and shrinkage. Now, once it is done, then elution can be carried out either in batch mode, or ion exchange slurry may be packed into a column and then eluted. Like in a batch mode, say once you have bound the analyte to the stationary phase, then sample could be decanted, and then elution buffer could be, first you can wash and then elution buffer could be put in the beaker, and batch elution, like say you can directly use 0.5 molar NaCl and let it be there with little gentle stirring, and then whole analyte will be eluted into that buffer like say 0.5 molar NaCl, and that could be decanted and could be used for further use, further applications. Or you can, so this is batch mode, or what you can do is rather than eluting in batch mode, you can pack whole thing in the column and then elute accordingly. In column adsorption, rather than batch adsorption, you can also do it in column adsorption. So, in batch adsorption, a protein is purified by adsorption of contaminants, which allows the desired protein to pass through the column without binding. Now, no concentration of protein results in here in column binding. Rather than batch mode, you can also do it by elution method. Now, here in elution method, it could be static ion exchange or dynamic ion exchange. Now, static ion exchange, protein is fully adsorbed to the bed and completely eluted by displacement into mobile phase using a small volume of strong eluent. Like I said, you can take 0.5 molar or 1 molar NaCl and you can elute the protein. Now, this is useful for concentration of protein from a large volume of sample, because what you are doing is you are eluting, though it is a strong eluent, but you are eluting it in a smaller volume. So, what you get is much concentrated solution of the protein. In dynamic ion exchange, separation of protein is achieved by their relative speed of migration through the column. So, what is done? All the sample components will migrate, but they will be separate depending on their relative equilibrium distribution or distribution coefficient we can say, or we can say relative affinities to the stationary phase. So, what will happen as you increase the ionic concentration, the molecules with different affinities will be eluted, and as I showed you earlier, they will be eluted in different fractions, and those fractions can be collected in fraction collector, and they could be observed on a spectrophotometer, or if your chromatograph contains UV monitor, then they could be observed in form of different peaks. There are three types of elution conditions which could be utilized. That could be isocratic elution. In isocratic elution, sample volume should be between 1% and 5% of the bed volume, and a long column is used with diameter to length ratio of around 1:20. So, the starting buffer is used throughout the separation. So, it is a isocratic elution where you are not changing the buffer as such, it is a one single buffer which will be eluting it, and it could be concentrated buffer or high amount of eluent might be utilized. Then there is stepwise elution. Now, in stepwise elution, a sequential discontinuous change in pH or salt concentration will be done. Like for example, say you can change pH sequentially, like you can first start with say pH. If I have to elute, I can start with say 7, then I could come down to 6, 5, 4 likewise, or I can go up in that order, or likewise salt concentration could be increased. Like for example, I can start with 100 millimolar salt concentration, then I can go to 150, 200, 250 and up to 0.5 molar NaCl. Now, column volume is determined by the exchanger capacity and the sample volume here. So, the sample should adsorb to 5 to 10% of the total bed capacity, and the column length is usually shorter. So, here it is a stepwise elution, and different fractions will contain different peaks, and those analytes having different affinities will elute accordingly. More affinity one will elute later on, and less affinity analyte will later first will elute first. You can use gradient elution also rather than stepwise elution. So, it is a continuous gradient here rather than step gradient. So, the composition of eluent, that is pH or ionic concentration, is changed continuously, and sample content should be 5 to 10% of the bed capacity ideally, and column length is usually 20 to 40 centimeter with a diameter to length ratio of not more than 1.5 1:5. The volume of the buffer required is determined empirically, and in too steep a gradient resolution will be lost, while too shallow a gradient will result in unnecessary dilution as long and long separation time. So, accordingly one has to choose the condition. The total volume of sample should be about 5 times the bed volume. These things are not hard and fast, and one has to really go through standardizing these values. So, in continuous gradient could be made by 2 pumps, and a gradient mixture could be used, where you can say start with 0 molar NaCl buffer and say 1 molar NaCl buffer, and accordingly you can have a gradient which could be too steep, or you can choose the gradient, it could be simple linear gradient otherwise. Now, if you see here, the chromatographic purification has been compared for a steep gradient and not somewhat so steep gradient, and you can see that here these two peaks, one is here only first peak is very clear, other peaks are not very clear, but here you can see two peaks are separating and other peaks are visible. So, that is very important, that separation of 4 peaks by ion exchange chromatography is taken here, but in steep higher steep gradient only one peak is very clear, other peaks are not so clear, but in shallower gradient of peak 2 and 3 also becomes clear, and existence of 4th peak becomes obvious. So, these are again as you go along doing these things, general guidelines are very clear, but one has to standardize its own conditions to get best results. Finally, once you have separated your biomolecules on column by combination of say cation or anion exchange chromatography, then the column could be reused many times. So, it has to be regenerated, and if you are not doing right away the next purification, you can store it also. Regeneration involves removal of tightly bound contaminants and conversion of the support to the required contourine form, ready for equilibration and protein adsorption. Removal of tightly bound protein is achieved, say, using 2 molar NaCl and then followed by an alkaline wash. For storage, antimicrobial agents are used to avoid microbial growth, and it could be high organic solvents are recommended, or sodium azide could also be used, like antimicrobial agents like sodium azide can be used. In certain conditions where antimicrobial agents cannot be used, then say 30% ethanol or other organic solvents could be used for this purpose. So, this completes our ion exchange chromatography section, and as we have gone through this particular section, I think I hope that you are able to understand that how ion exchange chromatography is performed. It is a very straightforward chromatography, but depending on the type of biological molecules to be separated, you have to worry about lot of different factors, and which include, say, conditions of the experiment, the stability of your analytes, the mesh size or porosity of the column, and the capacity of the column, standard conditions for running the column, kind of buffer you have to use, kind of eluents you have to use. All these things has to be taken care of. General guidelines are very clear, as it is very straightforward. If it is your analyte is positively charged, you have to use negatively charged cation, negatively charged column, if your and vice versa. So, but certainly one needs to standardize all the conditions, and then once for a particular analyte you have standardized the condition, that could be followed in your lab and purification can be performed. In the next lecture, we are going to discuss about another important chromatographic technique, and that is exclusion chromatography. Thank you.